This is a GCSE Biology presentation looking at stem cells. So to get started, try and come up with as many uses as you can think of for a paperclip. Pause the presentation, time yourself for 1 minute only, and make a list of as many things as you can think of. Pause now. What you should find is you should very quickly come up with a number, maybe 10 or so examples, but eventually find it very difficult. And the reason for that is you're making the assumption that the paperclip is a couple of centimeters in size and is made of metal. If you had used your imagination really well, you would have come up with lots and lots of other ideas if the paperclip had been 200 foot tall, if the paperclip had been made out of glass or made out of rubber, made out of gold, silver, etc., and come up with a lot more uses. In this session, what we're going to look at is stem cells. You're not just going to look at what a stem cell is, but we need to know where they're found, both in plants and animal cells. We need to know how they can be used to treat different conditions, and we need to be able to look at the risks and benefits of them and using them in medical research and medical treatments. When we say the term stem cell, what we mean is an unspecialized cell. That means it is a cell that has not been given a specific function. Those unspecialized stem cells can go through a process known as differentiation. Process of differentiation is what we do to turn a stem cell which is unspecialized into a different type of cell that is specialized. For example, a root hair cell or a muscle cell. You also need to know where stem cells are found. In a plant, stem cells are found in the meristem. In an animal cell, we find animal cells that are stem cells in the embryo. Just be aware that some stem cells can also be found in adult animals, but normally in the exam they'll refer to animal stem cells as being found in embryos. Now, we've already mentioned some examples of the cells that you can find inside plants and animals, and there's a separate presentation where you'll actually go through all the different types of cells that you need to be aware of, as well as the function and structure of each of those cells that make them specialized. But for now, just be aware that meristem cells, again, are linked to plants, and it's where stem cells are found in plants. But we have separate stem cells found in animals. And again, normally we talk about the embryo, but there are some adult ones. And you've got several examples of cells that can be differentiated into. So originally, it was believed that all stem cells came from embryos, but more recent research, we've been able to identify a number of adult-based stem cells found in a number of organs inside the human body. And what they're currently working on is taking these stem cells and changing the DNA inside these stem cells in order to change them into other types of cells. So stem cells can be used to treat a number of conditions. For example, paralysis caused by damage to nerve cells. Well, if we can take stem cells from somewhere else in a person's body, we can then change the DNA inside those stem cells and we can regrow the nerve cells that are damaged. And we can essentially cure someone of paralysis. In diabetes, you have a person who's not able to produce insulin. Again, if we take other cells from their body and we change those stem cells so that they become insulin producing cells and we inject them back into the person's pancreas, that person can then start to produce insulin and no longer suffer from diabetes. And because we're using a person's own stem cells, we don't have any problems with rejection of cells or rejection of tissue. In terms of plants, meristem stem cells can be grown very quickly, and it lets us produce large numbers of identical plants at very quick rates. And we have a number of uses for that. So for example, we can quickly repopulate species that are in low numbers, and we're also able to produce large numbers of genetically identical crops. So if we have a particular crop which has a particular advantage, for example, is resistant to a particular disease, we can then grow that in very large numbers very rapidly using stem cells. What you've got in front of you is a number of medical conditions and a list of cells that if we can take the stem cells from this particular part of the body, we can then modify that stem cell to cure and treat the relevant condition. Pause the presentation and try and identify which part of the body cells will be taken from for each of those conditions. When you're ready, you can restart the presentation and check your answers. So pause now. Well, if you were to treat Parkinson's disease, we would be taking cells from the brain. Spinal cord injuries, we would need nerve cells. If we're talking about heart damage, we'd need heart muscle cells. Diabetes, as I've already mentioned, would be pancreas insulin producing and insulin secreting cells. For burns and ulcers, we'd simply use any cells from the skin. And of course, anything to do with blindness, we would be looking at stem cells from the retina. What you've got now is a table showing you a whole list of advantages and disadvantages of using embryonic stem cells. What you have to do for each of the statements in a box is determine if you think it's an advantage or a disadvantage of using the stem cells. Pause the presentation, and when you're ready, restart and check your answers. Pause now. Well, the answers are as follows. Remember, you don't need to memorize every single one of these. You just need to have a couple of examples so that if you're asked in the exam what would be an advantage or what would be a disadvantage, you can give them some suggestions. So to finish off, should we be allowed to carry out research using embryos? What you need to do is come up with 2 reasons why you should and 2 reasons why we should not. Now remember, embryo stem cells are not the same as adult stem cells. So just be careful in the exam. If it talks about embryo stem cells or if it talks about adult stem cells, the advantages and disadvantages are slightly different. So for example, if we're talking about adult stem cells, you can't say that it would prevent a potential life from forming because that only applies to embryos.